Hi, I'm going to show you how to make a cheek cell slide. Probably not advisable to be doing this with many people at this time, but this is how we make a cheek cell slide using a light microscope. The first thing I need to do a cheek cell slide is to get some samples of my cells. So being that I am an animal, I have plenty of cheek cells and they look kind of like your standard animal cell that you draw, a bit like a wonky fried egg. So I'm going to get some cheek cells by doing this. And then I'm going to scrape that onto my slide, which is just made of glass. And I'm going to scrape that across like this. Delicious. Okay, this is going to go into some disinfectant and then straight into the bin. And then I'm going to put on a cover slide. This is my slide that has my cheek cells on it. And what I'm going to do is, because the light is going to shine through those and my cells aren't going to be particularly visible, is I'm just going to use a dye to make those cells more visible. When the light shines through, they are going to appear a lovely blue colour. I think I probably put a bit too much on there, but never mind. And then what I'm going to do is I'm going to put on a cover slip. And for that, I'm going to use a dissecting needle just to gently press this on to try and reduce the number of air bubbles that I'm going to get. I nearly always get them. There we go. I've got one really good one straight in the centre. And then I'm going to use a paper towel just to remove any of the excess dye. And that is just going to soak up the side there. The dye I've used is methylene blue. If I was doing an onion slide, I would use some iodine because that's a nice bright yellow colour. What I've done is I put my prepared slide onto the stage. Now the most important thing to do is to make sure it's clipped in place. It's not the most important thing, but it is a thing. And then make sure that the objective lens is on the smallest magnification and that the stage is as close as possible. When we use a microscope, we always start on the lowest magnification and work our way up. So when we're using our microscope, we're always focusing out. I've turned the light on at the bottom because it requires light. And I'm just going to start using the coarse focus until I can see an image. And then I'm going to use a fine focus so I can sharpen that image. Now, I'm now going to have to move on to a higher level of magnification. So I'm going to move around to the objective lens 10. So I'm now on 10 times 10, I'm on 100. And again, I'm just going to, before I start, I'm just going to make sure I'm on the closest focus and I'm going to start focusing outwards. Ooh, lots of cells. And use my coarse, then fine focus. Now we're going to make some plant cell slides. So you have to be able to do animal cells, and we normally do a cheek cell, but you also need to do a plant. So I'm going to go with one of the king of the plants. Not really. Good in most food though. We're going to do an onion cell slide. Now onion cells are really large. They're obviously not green, so they won't have any chloroplasts in them because they live under the ground, so they're not going to be doing photosynthesis. The leaves above the ground will be, so you won't be able to see any chloroplast structures, but you see a really, really nice uniform plant cell structure. So let's get cracking. What I need to do is to find my onion cell, so I'm going to cut my onion. Now the easiest place is we want to get one cell thick so we can get a really clear cell. So the easiest way to do this is to get a membrane. So here, oh, this is a really smelly one. Here you can actually see, look at that breakdancing on my screen. You can actually see a piece of the fine membrane. I'll just see if that can zoom. Stay still, can you zoom on that? No, it's not going to zoom. So that, I mean, is perfect. You can actually have fun with this at home when you're cooking, just see if you can get that membrane off. So I'm just going to show you here, look, this really fine sort of translucent membrane. It's actually easy to get off with your hands and you won't break it. Can you see that? So really fine. So this is one cell thick. And so I can use that to make my microscope slide. There we go. So let's get prepped, get all this out of the way. Nice, tidy work environment, Mrs. Corbett, good job. I'm going to get a slide. And again, this is a clean glass slide. Pop that on there. I don't need too much of this, so I need to get as like damaged free piece as possible. So I'm just gonna cut a bit. That'll probably do. I want to damage the cells as least as possible, so I've probably handled it. This one's really curly. Come on. There we go. Okay, now again, if I put the light through this on the light microscope, I'm not going to be able to see anything. So we give it a dye so that we're able to see the outline of the structures of the cells. They'll go into the cells, so I should be able to therefore see the nucleus and the cell wall. So I'm just going to put one blob of that iodine on there, and then I'm going to pop on a cover slip. And again, remember, cover slips are very, very fine, so we have to treat them very carefully, try not to get any fingerprints on them, so we always hold them at the edges, and then we need to try and get as few bubbles as possible. So I'm just going to hold it up with my dissecting needle. There we go. And again, I'm just going to remove any of the excess of that dye with a paper towel, just let that absorb up. I will stain your skin, so please be careful. So now I'm ready to use my light microscope again. Again, I'm not using an electron microscope. That would be used for very still, very small dead stuff. Okay, so we can use light microscopes because they have enough resolution to see these. So these cells are much larger than the cheek cells we saw on the previous video. And again, I'm going to be starting off on the objective lens on the lowest setting of magnification, and the eyepiece or the ocular lens is going to be stays the same the whole time. So it's times 10, and then the first setting will be times 4 magnification. This light at the bottom here adjusts to allow different amounts of light in, and that's can be called the diaphragm. Other words. I'm just trying to think if there are other words for any of these that we don't use necessarily. I think that's probably it. Let's get this on and see. So I'm going to start with the lowest magnification, pop it on the stage, put it on the clips, put the light through to ensure the light is shining through the cells so you can see the structures. Boom. And then I'm going to start magnifying the same way. Again, always focusing outwards using the coarse, this side, the coarse, and then the fine focus. That's really important on exam questions, to make sure you mention both of those and that you use coarse first. And that's it. You can have a crack at this when we get back to school. Bye, science fans. This is really hard to focus because I'm trying to focus and I've got one of these phones with like 3 cameras. But this is what the onion cells look like. It's really funny, I'm holding this dead still, but it looks like it's dancing a little bit. But you can just see the outline of the cells. So that is on magnification 10 times 10, so on 100. I've lost it again, there it is. Pretty awesome. There it is, fantastic. Onion cells, look at that uniform shape. You can see the cell wall, you can see the nucleus, you can see the cytoplasm. But that's about as much detail as you're going to get on a light microscope.